Okay, so what we're going to do is go through a wee short mitosis, all right? So if we look at this first picture here, right, where do I normally find chromosomes? I normally find chromosomes inside the nucleus. So one of the things that's happened here is the nuclear membrane has disappeared. Okay. Again, if we now look at this, the other thing that's happened is DNA replication has occurred. Each chromosome at this point, so if we look at that one chromosome, what's our one chromosome made up of? So one chromosome made of two identical chromatids. That's what we've got here in this first picture, okay? If we look at picture number 2 here, so stage 2, what's happened now? These chromosomes lined up along equator. And our wee spindle fibres, so look at these are our wee spindle fibres have formed. Okay, so that's what's happened in our picture 2, right? Our third picture, have a wee look at this. What's happened here is these spindle fibres on this side have contracted up this way, these spindle fibres have contracted that way, and what's happened is the chromatids have been pulled apart to opposite ends of the cell. Okay, so that's what's happening in that picture. Okay, we'll look at the next one. So our stage 4, what's happened here? New nuclear membranes have formed around the chromosomes. And then what's happened here? Look, our cytoplasm is divided. And what do I have? I have two genetically identical daughter cells. These daughter cells have to be genetically identical to the original cell and each other so that they carry out the correct function, so that they function properly. Because if I was missing any genetic information, then I can't carry out the correct function, okay? So our nuclear membrane is disappearing. Our DNA replication occurs. Each chromosome is then made up of our two identical chromatids joined at our wee centromere. Now that's extra information, we don't need that. So our wee chromosome made of two identical chromatids. Okay. What's happened now is our chromosomes, they've lined up along the equator of the cell. And our wee spindle fibres, look at our wee spindle fibres, they've formed. Okay. Stage 3, what's happening? Look at this. I have separated the chromatids. I've pulled the chromatids apart to opposite ends of the cell. Next, new nuclear membrane forms around our chromosomes, cytoplasm divides, and I now have two genetically identical daughter cells. Why is that important? So that they can function correctly. Now, I just want to go through this again with a picture that looks a little bit different. So, if we look at these two pictures, what's happening here? Nuclear membrane disappears. What else is happening? Now, see in this picture how we start to see the chromosomes? That's because DNA replication is occurring. And see when we do DNA replication, each chromosome made of two identical chromatids. Okay, what's happening in this next picture here, right? Chromosomes have lined up along the equator. We will also have our wee spindle fibres forming from our poles. Okay, next picture, what's happening here? The chromatids are being pulled apart. They're being pulled apart to opposite ends of the cell. Look at this last picture here. What's happening now? New nuclear membranes are formed around the chromosomes. And cytoplasm divides. And what I've got to think here is, what do I have? I now have two genetically identical daughter cells. And these genetically identical daughter cells, they're genetically identical to each other and the original cell, right? Why is that important? So that they can function correctly. Okay, so let's go through some questions now, right? Now what I'll do is I'll pause at the start and try and give you a wee bit of time. So what you can do is just pause the video, read through the question, you can answer it yourself, and then if you press play again, you'll have me talking through it, okay? So a cell with 10 chromosomes divided by mitosis. Which row in the table identifies the number of daughter cells produced and the number of chromosomes in each daughter cell? So the information we're being told there is that I've got 10 chromosomes, right? Now my daughter cells, I will start off with one cell, right? What is going to happen is I'm going to divide into two daughter cells, so I need to have either C or D, right? If this cell has got 10 chromosomes, I need to be genetically identical. Answer is C. Okay. Next one. Again, wee bit of time, you can pause. Diagram below represents a cell in the early stage of mitosis. State the number of chromosomes present in this cell. So all we've got to do is count. Look, there's one chromosome, 2, 3, 4. So there are 4 chromosomes in that cell, right? Look at each one of these chromosomes. Each chromosome is made of 2 identical chromatids. Okay, so that's why they look almost like funny wee X's. State how many chromosomes will be present in each of the 2 daughter cells produced. So remember, daughter cells, they've got to be genetically identical to each other and the original cell, so 4. Okay, identify structure X here. So what is this little dude here? Spindle fibre. What would happen in stage D here? So what I've got to look at is C. What's happening here? Chromatids are pulled apart. So I've got to think about what's going to happen next, right? See, once the chromatids have been pulled apart, what do I want to do? I want to chop the cytoplasm in half. So cytoplasm divides. The other thing that I could think here is, will I need new nuclear membranes around the chromosomes? So there's my 2 option, right? Cytoplasm divides, or new nuclear membrane forms around the chromosomes. So following phrases describe stages in cell division. So we have this stage P, chromosomes line up along the equator. Q, nuclear membrane forms, cytoplasm divides. R, chromatids separate, move towards opposite ends of the cell. S, each chromosome doubles itself, appears as coiled threads. Now we've got to put these in the right order, right? Now these aren't worded the way we would want to word them. We would be wanting to look for something about, DNA replication occurs and each chromosome is made up of 2 identical chromatids. Now that's not there, but look, chromosome is doubling itself. So let's start with S, okay? Now what do I want? I want my nuclear membrane disappearing. There's nothing there. I want my chromosomes lining up along the equator of the cell. There we go. So P. After my chromosomes line up along the equator of the cell, I want to separate the chromatids to opposite ends of the cell. So there's your separating the chromatids to opposite ends of the cell, R. Their new nuclear membrane is going to form, and then cytoplasm divides, Q. Okay. This question down at the bottom, it says a cell divides every 20 minutes. How many cells would be produced from one original cell at the end of 2 hours? So if you think about this, I have one cell here at the start. After 20 minutes, how many cells will I have? I'll have 2 cells, right? So at 0 minutes, I'm going to have 1 cell. After 20 minutes, I'm going to have 2. After 40, I'm going to have 4. And I literally just keep going on here. Every 20 minutes, it doubles, right? So the 60, then I want to go 80, 100, 120. Now that takes me to my 2 hours. So after the first 20 minutes, I had 2 cells. After 40 minutes, doubled again, 4, doubled again, 8, double again, 16, double again, 32, double again, 64. I will have 64 cells at the end of my 2 hours. The diagram below shows one of the stages of mitosis in the root tip of a plant. Which of the following statements describes the stage shown? So if we look at A, it's saying chromosomes line up along the equator. Does that look like that's what's happening? B, daughter cells gather at the ends of the cell. C, chromosomes become visible as pairs of identical chromatids. D, spindle fibers pull chromatids to opposite ends of the cell. There we go. That's what's happening. Our chromatids are being pulled apart to opposite ends of the cell. Here we've just got to match up the pictures, right? So what's happening in this picture here? Look at this. The chromatids are being pulled apart. So where's my chromatids being pulled apart? Here we go. Here, what's happening here? Look, my chromosomes have lined up along the equator of the cell. There we go. All right. Question down here. How does mitosis ensure that daughter cells will be able to function? So how is I making sure that they could function correctly? They are genetically identical to, and we could say each other, but most importantly, to the original cell. Whatever job that original cell does, if I'm genetically identical to that original cell, I can function correctly. So here, chromosomes become visible as pair of chromatids are stage 1. Stage 3, pairs of chromatids attached to the spindle near the middle of the cell. Okay, so I've got to decide what happens in stage 2. So look at stage 1. I've got my chromosomes are made up of my 2 identical chromatids, and then here I've got the spindles being attached to these chromatids near the middle of the cell. So what's going to have to happen in the middle here? Chromosomes have to line up along the equator. Now, if I also said nuclear membrane disappears, that is fine, but I must say chromosomes line up along the equator, because my chromosomes would have to line up along the equator before I can get them to attach to the spindles. Okay. Stage 4 is missing, but I've then got this stage 5, daughter chromosomes gathered together at the ends of the cell. So what has to happen in between? Chromatids are pulled apart. To opposite ends of the cell. So daughter cells produced by mitosis each have the same chromosome complement as the original cell. Why is this important? So why is it important that they're genetically identical? So that they can carry out the correct function, so that they can function properly, so that they can function correctly. All we've got to do here is put these in the right order. So if we have a wee nosy first, what we've got to think is, what do I want to start with? My DNA replication, each chromosome is made up of 2 identical chromatids. So there we go, number 1. After this, right, what's going to happen? Nuclear membrane disappears, my chromosomes line up along the equator of the cell. 3. After my chromosomes have lined up along the equator cell, what's going to happen? I separate the chromatids to opposite sides of the cell. I pull apart the chromatids to opposite ends of the cell. Then cytoplasm divides. So we are 1, 3, 2, 4, which is A. Okay, which of the following does not involve mitosis? All right. So if we go through this one by one, right? Repair of tissue. Would I need to make new cells? Yes, I would. All right. Maintenance of the diploid chromosome complement. So see our cell right at the very start, if that has 2 sets of chromosomes, would my 2 daughter cells both have 2 sets? Yes, because they would be genetically identical. So yes, right? Growth of tissue. If I was growing tissue, would I need more cells? Yes. Protein synthesis has nothing to do with mitosis. Table describes some stages which occur during cell division, but not in the correct order. The first stage has been given. Identify the third stage by writing the number 3 next to its description. So I think the easiest thing to do here is just work through, right? So chromosomes shorten and thicken. Now we wouldn't say that, but what we would say is we would say, oh, our chromosomes, we've done DNA replication, our chromosomes are made up of 2 identical chromatids. Now, I would be looking for nuclear membrane disappearing. That's not there, okay? What I would be looking for next is chromosomes lining up along the equator of the cell. So there's number 2, right? Chromosomes line up along the equator of the cell. What do I need to do now? I need to separate the chromatids to opposite ends of the cell. There's number 3, okay? Nuclear membranes are going to form around the chromosomes and cytoplasm divides. See the 2 of these? It doesn't actually matter what order you do them in, right?